Hello, and welcome to this discussion of some of the latest data in the field of lupus. I'm Vibeke Strand. I'm a biopharmaceutical consultant and adjunct clinical professor in the Division of Immunology and Rheumatology at Stanford University School of Medicine in the US, California to be complete. Today, I'm delighted to be joined by someone who you'll all be familiar with, Dr. Joan Merrill, Professor of Medicine, Arthritis, and Clinical Immunology at the University of Oklahoma Health Sciences Center in Oklahoma City. Welcome, Dr. Merrill, and thank you for joining me to discuss the results of your phase 2 trial of Iberdomide in lupus.
Hi, Vibeke.
It's great to talk with you, Joan. So before we take a closer look at your results, please can you give us some background on the Iberdomide program for the treatment of lupus?
Sure. Iberdomide is an inhibitor of two key transcription factors that help regulate the immune system. And they have Greek names, Ikaros and Aiolos. Both of these transcription factors kind of get things revved up, headed towards the type 1 interferon pathway and the manufacture of IL-2, both of those being key players in lupus pathology. So Iberdomide actually binds to something called cereblon, but when it does that, it actually allows these transcription factors to sort of be loose and get ubiquitinized. So ubiquitin is put on them, and that leads to their destruction. And there was an earlier small study of Iberdomide performed at Celgene that showed some promising data, which then led the way to this phase 2 trial.
Thank you. So with your experience in trial design, because we know you have a lot, I'm sure there were many factors you took into consideration for this phase 2 trial of Iberdomide.
Well, I know that you've been very involved in trial designs yourself, and you know what a very complicated and problematic field it is, particularly for lupus. In 2018, a group of people got together and came up with some kind of a roadmap for things we could try to get these trials to be more interpretable. And this particular phase 2 trial used some, but not all of those suggestions. So one of the things that it really did not do is have a strongly, what we call a strongly encouraged taper, because we don't like to call it mandated. Obviously, if you're tapering steroids in a patient with lupus, you don't want to do it if it's going to get the patient in trouble. So doctors always have the right to override a taper. But I guess what's mandated in this trial design that we didn't do, but has been behind some of our recent successful trials, is really making sure that the doctors would at least consider steroid taper. And if they decide against it, that they would give the reasons why and really have that subject to discussion. So that's a part that was not used, but the other parts were used. And this involves having experienced lupus trialists, experienced lupus clinicians, not just trialists, look at each patient before they enter and ask themselves 3 questions. Does this patient have lupus? If the patient has lupus, are the current disease manifestations that are making them enter in the trial, the scoring of these instruments, is that due to active disease and not damage and not some collateral illness?
Right.
And then finally, is the patient, stable enough to come in a trial? And that was taken very seriously in this trial, as well as expert monitoring of all the scoring throughout the trial. Right. And I do think that's helping us to support more. I know, Vibeke, you've been engaged in this kind of thing yourself, so you know what I'm talking about.
Absolutely.
So I think it is helping us get more interpretable trials. And so I think I would say those are the factors that really may have led to a successful result here.
Well, I think I strongly agree with you. So the 0.45 milligram dose stood out with a significant difference in the SRI-4 and clinically meaningful improvements in S2K scores. Mm. The other secondary endpoints were not met, and there wasn't statistical difference between the other doses of Iberdomide, but I suspect that you didn't expect that, at least with the lowest dose. So what does this really mean for going forward with this product?
Well, there are two things that are sort of behind the data that were not really brought out very strongly in the paper as published. You know that the journal that it was published in is a very respected and very conservative journal. So you have to be very careful what sorts of things that you claim for your drug. And one of the things that I know you know is that in a clinical trial, if you don't meet your first secondary endpoint at the predicted statistical significance, nothing counts. And so again, and several times we were required to say we did not meet our secondary endpoints. But I would also point out that this is a phase 2 trial. And in phase 2, we do look at exploratory or secondary endpoints that come down the line after the first ones aren't met. Not to make any claims, absolutely not. And the journal is correct about that. But to make hypotheses about whether it's worth doing a phase 3 trial, and if so, how to design them. So there were some endpoints that were met. Not met, I shouldn't say that, because we're not allowed to say they're met because they're high down the hierarchy. But there were some that provide interesting hypotheses. One was if you just looked at the SRI-4 result only in the subset of patients who are more ill, defined as a SLEDAI score greater than 10. There was a bigger difference between drug and placebo than with the overall population. Now, what's important about that, I think, is that if you think a drug might work, if you try the sicker patients and it works even better, usually that has to do with the placebo group doing worse, but you know, at least it suggests a robustness to the drug that it can also help the sicker patients. Also, it showed a signal with SRI-4 with an additional requirement for steroid reduction. Now, there wasn't a lot of steroid reduction in this trial, unfortunately, because of the design, but that was a sort of a hint. And then also resolution of arthritis or rash by using just the SLEDAI, which means very high bar. They pretty much had to go away or they would have counted. Yep. So, so those, you know, showed an efficacy signal. So I think the data for generating hypotheses is a little better than the data you can make claims on.
Well, I would agree with you. So tell me something. Are any of the other doses going to be examined going forward, or is it just going to be the 0.45?
Well, there wasn't. I actually don't know the plans of Bristol Myers Squibb, who owns this drug now, but I will say that I would like to see more study of all of these things. One of the other issues is that the 0.3 milligram dose had a large number of dropouts. And although that may be telling us something, it may not be telling us anything because it's such a small trial. So I think that we shouldn't put that to bed yet.
So I would agree with you. Being a small trial, the small number of patients, it's really good that you even got a statistical difference with the primary endpoint with the 0.45 milligram dose.
Correct.
So what about the safety profile? There were more adverse events in the 0.3 and 0.45 doses.
Yeah, there were, but nothing out of line with what you see in lupus trials. We didn't get a big signal from, you know, again, anytime we talk about interfering with the type 1 interferon pathway, we always worry about zoster reactivation and things like that. Right. And like anifrolumab, also, we're not getting a very scary signal from any of this stuff. No. There were more infections, and you expect that pretty much in line with other lupus trials and things we would expect.
Yeah, but they were mostly URIs and UTIs.
Absolutely,
and so
absolutely.
That's not very worrisome to me either.
Correct, correct.
So the changes in the specific populations, those with the Aiolos high and the interferon 1, type 1 interferon signature high.
Yeah.
Also because took a look at the Lipsky article about the specific biomarker data. So what do you make of all of that?
Well, look, if this is a drug that interferes with what Aiolos is going to do, and if what Aiolos is going to do is activate the type 1 interferon pathway, it makes sense that it would work even better in people who come in with high Aiolos or a high interferon signature than in patients who don't have that. In fact, it worked better than the overall population with whatever these patients had, upstream or downstream of where it would act. And very intriguingly, I think, is that when phase 2 anifrolumab showed that they had a larger effect size in patients who had a high interferon signal, they didn't really have more people responding to the drug. They had less people in the placebo group. So they could have just been looking at a biomarker for sicker patients, not necessarily for their drug. In this case, it looked like, and again, this is small early data, but it's intriguing. It looked like there were a much larger proportion responding to the drug in these groups. So that's kind of cool. And I think that's what we want to study more about. And the Lipsky paper also pointed out that there was a decrease in B cells and a decrease in plasmacytoid dendritic cells. And this is stuff we would expect from this. And so that's, it kind of gives you a rationale that this makes sense. The data, whatever we're looking at, it's early. We can't hang our hats on it. We can't even make any claims for any of those secondary endpoints because they broke the statistical law. But we sure can see a pulled together rationale that seems to make sense to generate a strong hypothesis. So I do hope they go into phase 3.
Well, I share that hope. So the Ikaros high population was pretty small, didn't really see much there. Yeah. But you did see increases in IL-2 and Treg. So that would be supportive of all the findings.
Correct.
So can you explain to us the clinical relevance of this trial? I think you already have, but maybe you want to just highlight that again.
I think the clinical relevance of this trial is, in a way, if we're doing a laboratory where we're taking all the good ideas about how to do phase 2 trials, here's one that took one of the big ideas and didn't take the other. It still seems to have succeeded. So we can begin to deconstruct our ideas and see, at least circumstantially, what works and what doesn't. So we did okay here with one of those big ideas. Of course, there's no placebo group when you do trial design tests. You'd have to do another trial just like it without doing that. Right. So that's the first thing. It sort of helps us in our figuring out how we do these phase 2 trials to get interpretable results. The second thing I think is that, these biomarkers and this approach of trying to find a subset more likely to respond is here to stay. I think it's got major advantages to us selecting drugs for our patients in the future, but the disadvantage, of course, is that drugs may become inaccessible to patients who don't fit the bill, who might have responded, because some people might have. So I'm hoping that we can work with regulatory agencies in the future and try to come up with a compromise that maybe if it's not the first line drug for the non-propitious patients, it could be down the line something that could be tried for them.
So something is specific to me. You had the facet fatigue in there, and it didn't really show a lot of differences, but again, small numbers of patients. Did you have any other patient-reported outcomes included?
I don't have any data for you right now on that. But yeah, we were pretty disappointed about that because in some trials, the facet fatigue works really well. It was a pretty short trial. This is a 6-month report. I think we just want to keep seeing what happens with that.
I think most of the PROs that we look at lupus, with the exception of global, we have too small patient numbers in phase 2 to look at them. And really the data with anifrolumab was statistically significant and quite positive when both trials were combined and we looked at responders.
I still like the SF36 and
I'm glad you do.
Well, you found an MCID for it, and I think that's really important. I always tell people, I think we should use one where we kind of know what a meaningful difference is.
Yeah, I have to agree with you. And, I was very impressed. Well, the belimumab data was positive in responders by SRI-4 versus non-responders, but anifrolumab data had also the lupus qual in it, and everything was statistically significant, including facet. And so I hope we see more of that and also get a patient global, because it does tell us how our patients are doing. Anyway, what do you think are the next steps in this clinical trial program, since you actually don't know? What are you hoping for?
I hope they'll continue studying it. And if I owned the world, I would do a phase 3 trial. I think we're ready, and I think we have a very good evidence-based guess about how best to do it. The secondary endpoints that were not met were ones that I don't like anyway. Deconstruction of the SRI-4, I just don't see the point to it. So you met the SRI-4, why are you looking at the components of the SRI-4? Also, joint counts, they're okay in lupus. They really don't belong at the top of the secondary endpoints because they're hard to meet in lupus. So the CLASI was a disappointment, but they were looking at patients who came in with 10 or more, and I think that was just an underpowered study. So there are a lot of reasons why those first secondary endpoints didn't work.
Yeah, that makes a lot of sense to me too. So I just wanted to say thank you again for your time. And if you'd like to know more about this paper, you'll find a summary slide deck on the Lupus Forum website, lupus-forum.com, where you'll also find a wealth of publication summaries, podcasts, Congress materials, and therapy essentials. Please subscribe to this podcast on iTunes or other podcast media and let us know what you think. Thank you.